Seventh graders, part 4 of the DNA lab stations. This is one of my favorites. It's called Who Ate the Cheese? So this is where you guys again kind of get to use DNA, except you guys get to see the process a little bit more firsthand and kind of learn a little bit more how forensic scientists would use DNA to solve a crime. So your objective in this simulation, you will examine crime scene evidence to determine who is responsible for eating the Queen's special imported Lindenberger cheese. Yes, the stinky kind. You will model the process of electrophoresis and DNA fingerprinting. Okay, so this is a theft processed by Chief Wiggum. The complaint status, pending DNA results, and the other officers involved are Officer Legace. Okay, this is a rare Lindenberger cheese. It's worth about $12,000 for the wheel from Queen Elizabeth. Okay, method of entry for the burglary, unknown. No evidence of force on doors or windows. Okay, narrative. The cheese was allegedly stolen from the Queen's sitting room the night before the grand ball. The cheese was listed as a gift from the Manchurian diplomat. Officer Lee Gase dusted for fingerprints and found none on the table or doors. The maid claimed that they had been wiped clean earlier. The wheel of cheese was on a platform in the sitting room and half of it had been eaten. We took pictures of the half-eaten cheese and sent it to the lab for further tests. So really they only ate like $6,000 worth of cheese. It's totally fine. Edna Ensign, the lab technician, said that the saliva samples could be taken from the teeth imprints of the cheese that was left behind. Let's learn about our suspects. Suspect number 1, Princess Deba Elix. Description of suspicion: the princess was seen entering the sitting room earlier in the evening. She is well known for her love of cheese. Suspect number 2, electrophoresis. Electra was recently involved in a relationship with the Manchurian diplomat that sources say ended badly. Her motive may have been to sabotage the diplomat's gift to the Queen. Suspect number 3, Adenine. Ada was the maid in charge of cleaning the sitting room. She had access to the cheese, but does she have the motive? And finally, suspect number 4, Gene Tix, or genetics, if you guys want to read it differently. Gene is the leader of the local cheesemakers guild, and he may not have wished for Queen Elizabeth to have cheese from anywhere but his own guild. So let's figure out who did it. Okay? So what you guys will see is after receiving the package with the plastic bag marked crime scene, the DNA was extracted. Because the sample was so small, again, we talked a little bit about this, it's not a lot of DNA when you guys get these samples, so you got to really replicate that DNA so you have enough to work with. So the DNA was amplified using polymerase chain reaction, or PCR. We isolated the DNA from 4 suspects and compared them to the crime scene DNA using a DNA restrictive analysis. So you guys are going to be the actual chemicals that do this DNA restrictive analysis. So what you guys have here is, as we know, DNA is double-stranded. Okay, so you guys have double-stranded pieces of DNA. We have the crime scene DNA. This is what we found in the saliva on the cheese. We have suspect 1, which was Princess Deba Elix. Suspect 2, which was electrophoresis. Suspect 3 was the maid, Adenine. And suspect 4 was the cheesemaker guild, Gene Tix, I believe. Okay, or genetics. So what you guys are going to do is you guys are going to look for, does it tell it up here? No. Where did I put the directions? Okay, you guys are going to look for the code CCGG. Because our DNA is double-stranded, you guys only need to look at the top line, okay? These, again, both these lines just kind of represent your 2 strands of DNA. So we don't have to look at this second strand of DNA at all. So let's go ahead, we'll do the crime scene together. So what you guys are going to do is you're going to find the code CCGG. Here's CCGG, and you're going to draw a line after it. You guys are going to go down the whole length of your DNA until you find CCGG. You guys will definitely probably find it more than once, not always the case. And then you guys will draw a line. Basically what you guys are doing is you guys are doing the part of this chemical called DNA restriction enzyme, which basically is kind of like a little chemical set of scissors that identifies a certain code and then it cuts the DNA in that areas. So we can examine how big these chunks of DNA are. So we've got one last one right there. So basically you guys being that chemical just cut the crime scene DNA into 4 different pieces. The first piece is 1, 2, 3, 4, 5, 6, 7, 8, 9 nucleotides long. This one is 25 nucleotides long. This one I believe is 11. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. I was wrong. 17. 17, and then this last one is 2. Okay, so that's how many pieces of DNA we cut our DNA into for our crime scene DNA. We had one piece that was 9 bases long, one was 25 bases long, one was 17 bases long, and the other one was 2 bases long. So what you guys are going to do then is after you've cut each strand of DNA, you guys are going to go to slide 13, and you guys can see we have crime scene DNA right here. What we're going to do is we're going to draw a little line for each of our pieces. So our first piece was 9. So we're going to go ahead about where 9 is, and we're going to draw just a line, okay? Our next piece of DNA for the crime scene was 25. So we're going to go up here to about 25, be as accurate as you can, and draw a line. Then we had 17. So we're going to draw a line here about 17. Oh, that's a solid line. Oh, that's a solid line. And our last one was 2. Okay, so if we were to run these through this thing called gel electrophoresis, the heavier pieces are gonna stay the closest to the well up here, and the lighter pieces are actually gonna travel further. So we're able to kind of separate the DNA based on how heavy they are. Okay, our heaviest pieces are 25 because it was 25 base pairs long. Okay, that one's gonna be closest to our little mark of crime scene DNA. Okay, you guys can see there's already one done on suspect 4, so let's go ahead and take a look at suspect 4. Okay, you guys are gonna do the same thing. We're only looking at the top line of DNA. You guys are going to go through, you're going to find CCGG, you're going to draw a line and count. CCGG, draw a line and count. And CCGG, draw a line and count, and then you guys will graph that. Okay, so you'll count how many base pairs, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So that's where that first bar is already kind of done for you guys. So you can see here's 12. Okay. So go ahead, you guys can finish counting for suspect 4. Count this chunk, count this chunk, count this chunk. It's split into 4. The other ones won't necessarily split into 4 chunks like the crime scene DNA and suspect 4 did. So go ahead, finish up suspect 4, graph the other 3 bars, and then go through, and again, you guys are just going to look at the top line of each of these. And find CCGG, draw a line after it. And then count each of those segments and graph them right here, okay? Then what you guys will be able to do is, all right, if this is our crime scene DNA, compare them to our 4 suspects, and then you guys should be able to answer the question, who stole the cheese? So highlight which suspect you guys think is the culprit, and how do you know? Okay, that concludes part 4, who ate the cheese.